Okay, I'd like to welcome everyone to today's webinar. My name is Yichao Rui. I am a soil scientist at Rhodell Institute. Also serve as the research director of the farming systems trial. Here at Rhodell Institute, I support all soil health related research and education. Today, I'll be talking about an interesting topic that everyone is interested right now: regenerative agriculture and soil carbon dynamics. We will be discussing where agricultural systems can be designed and managed to regenerate soil carbon, and how the recently proposed soil microbial carbon pump conceptual framework can be used to explain the mechanistic processes of soil carbon dynamics under regenerative agriculture. So, before I get started, just some housekeeping rules. During the presentation, I cannot hear you, but hopefully you can hear me. So if you experience any difficulties, please call Maria Pope, our Director of Communication, on 610-683-1481. Please use the Q&A function to send your questions through. I'll respond to each of your questions at the end of the presentation. And as usual, this webinar will be recorded and uploaded to our website and social media, including our YouTube channel, so you can watch and share later. So for those who are not so familiar with Rhodell Institute has been widely recognized as the birthplace of organic agriculture in the United States. Founded by J.I. Rhodell in 1947, Rhodell Institute has been growing and leading the organic movement in the United States and in the world. Our headquarters is located in Kutztown, Pennsylvania, about 2 hours from New York City, 1 hour from Philadelphia. But we also have 3 regional resource centers in Iowa, Georgia, and California. And very excitingly, we just opened a new resource center in Italy. So we're expanding our global education and outreach efforts. So if you're from or nearby these areas, please come over and visit. And we believe that healthy soil equals healthy food and healthy people. So soil health is really the foundation for food production and human health. But intensive agricultural system or agricultural practice can cause decline in soil health. So the decline of soil health is something that can threaten the long-term food production. So when soil is degraded, we believe that it's not enough to sustain soil health. We need to be actively regenerating soil health. That's why in the 1980s, Robert Rhodell, J.I.'s son, started to use the term regenerative agriculture to mean a kind of agriculture that goes beyond sustainable. So regenerative agriculture aims to not only sustain resources, soil health, but also actively improve them so as to build a strong foundation for long-term food production. Now, I'm sure that many of you are becoming more and more familiar with the term regenerative agriculture, because in the last 5 years, it has really caught many people's attention and has become a buzzword in the media. So I always hear people ask, what's the definition of regenerative agriculture? What's the difference between organic, sustainable, and regenerative agriculture? So I want to talk a little bit about the definition of regenerative agriculture. So we use the term really because we think that soil is the foundation. We need to keep improving soil. And the difference between organic, sustainable, and regenerative agriculture is that now organic agriculture is strictly certified operation. For example, in the United States, it's certified by USDA, and in other countries, also organic, also are certified by other governmental agencies. And the term sustainable agriculture was used very widely in 1990s or early 2000s. So it sounds like it can mean a lot of things, but actually it sounds very vague as well. So it's not pointing to any direction that where agriculture should to aim to. Regenerative agriculture instead, it's pointing to a direction that where agriculture should go to, right? For example, we believe that we need to keep regenerating soil health, soil organic matter. That's a journey. So if you are a conventional corn and soybean farmer, but you have started adopting cover cropping and no-till, so you started your journey of regeneration. But you can keep moving further to the direction of regenerative agriculture. So regenerative agriculture, it's a journey, it's not a destination. So that's what we believe, what regenerative agriculture is. So as I just said, improving soil organic matter or soil organic carbon is a focus of regenerative agriculture, because healthy soil is really the foundation for many important ecosystem services, including plant production, water quality, human health, and climate. And soil health means not just carbon, of course, not just carbon. It also means the physical, chemical, and biological aspects of soil health. For example, for a healthy soil, we always want a healthy soil structure. We want the 50% of the soil space is filled with water and air. So that's a pore space. That's a physical structure we want. And for chemical properties, we want soil to have good buffering capacities. And we also want soil to have, improved or balanced biological interactions, biodiversity. But among all the soil properties, soil organic matter is really central to many functions, most of the functions that soil perform. For example, with improved soil organic matter, we usually see an improved water holding capacity, improve the pore space, pore structure of soil health. And we also see increasing capacity of soil to supply nutrients for food production. And of course, with more organic matter being stored in the soil, we have less carbon emissions into the atmosphere, which can help to mitigate climate change. So improving soil organic matter content or soil organic carbon content is really the focus of regenerative agriculture. It's something measurable. When I say soil organic matter and soil organic carbon are usually used interchangeably, because we know that soil organic matter contains carbon, nitrogen, phosphorus, sulfur, and many other elements. But usually carbon makes about 52 to 57% of the total soil organic matter. So soil organic matter and soil organic carbon are usually used interchangeably. Okay, so what can we do to improve soil organic matter? That's the million-dollar question, right? So if you look at the principles of regenerative agriculture, you can see that many of the principles, including eliminating chemicals, diversifying crop rotations, maximizing living cover, using natural fertilizers, and reducing tillage and soil disturbance, can all contribute to improving soil organic matter. But they may contribute to improve soil organic matter differently. They work from in different aspects. That's really because soil, carbon in the soil, carbon content in the soil, carbon stock in the soil is really about the balance between input and output. So if we have more carbon coming into the soil than carbon being released from the soil, then we see net carbon sequestration, and we can say that soil is a net carbon sink. But if we see more carbon going out to the atmosphere than carbon going into the soil, then we see our carbon, a net carbon release. So soil is the carbon source, which in this case, soil and agriculture contribute to the climate change by producing more greenhouse gas emissions, which is estimated about a quarter of the greenhouse gas emissions right now by IPCC. So because carbon content or soil carbon sequestration is the balance between input and output to sequester carbon in the soil, we can focus on different strategies. Strategy one is to maximize carbon input. So what can we do to maximize carbon input? So we can plant cover crops, right? Cover crops are planted in between the cash crops. For example, in the winter time, a lot of times farmers leave the soil bare, uncovered. But if you plant cover crops and let them grow in the winter time, although they grow slowly, but they still can perform photosynthesis and fix carbon through photosynthesis, right? And put carbon into the soil. We can keep the residues instead of taking them all of them away after the harvest. We can use organic amendments such as compost and manure. We can also plant perennials because perennials can grow all year round for several years. So even in the wintertime, like cover crop, they can still grow and perform photosynthesis and fix carbon. So by adopting these practices, we can maximize carbon input, right? And the second strategy is to minimize carbon output. How can we achieve that? So carbon output can be minimized by avoid frequent and intensive tillage. For example, if we till the soil 3 times a year, and we use a very aggressive mobile plow every year, then we will introduce more oxygen to the soil. We will expose the previously protected soil aggregates, then cause soil carbon to be released through microbial respiration, right? So we can reduce tillage. We can adopt no-till or reduce tillage practices. Or we can, like the previous strategy, we can adopt perennialization. We can plant more perennials, so we don't have to till the soil every year. There's one other strategy that was overlooked for a long time, but it started to show a lot of promises. So this is to build stable organic carbon, build stable organic matter. What does that mean? So that means we can promote the formation a type of organic carbon that can persist longer in the soil, that can stay longer, can be more stable, so they can contribute to improve the soil carbon sequestration. So how do we achieve that? So this is something we're going to focus today in today's presentation. So that actually leads to the question, what is soil organic matter? What's the nature of soil organic matter? So soil organic matter by its definition is the organic fraction of the soil, including plant, animals, and microbial residues at all stages of decomposition, That means these organic materials come into the soil from the raw material, from plant litter, roots, or leaves, or animal dead bodies. They will be converted and to the soil organic matter through different processes of decomposition. And the organic matter is one stage of the decomposition where the organic matter is stored in the soil. So actually, our understanding of the nature of soil organic matter has experienced a paradigm shift in the last maybe 10 years, in the last decade. We really have evolved our understanding of the nature of soil organic matter. You probably have heard of the term humus. Humus, right? Humus was used by many people to mean the stable soil organic matter. So that was actually the traditional or historical understanding that we believed the humic polymers, those long chain structured compounds, they were actually the soil organic matter. People believed that soil organic matter was formed as those recalcitrant, very chemically complex polymers, compounds. They they form the soil organic matter, or the so-called humus, through condensation or humification. So that's the result of recalcitrant materials, right? But now, because of the advancement of technology, the direct high-resolution observation on soil, in soil aggregates or on soil mineral surface, only found simple molecules, very simple structured compounds, not complex polymers, form stable soil organic matter. That's really contradictory to our previous understanding that soil organic matter was recalcitrant and chemically complex. So organic matter now, we start to realize it's actually very simple structured material, very simple structured molecules, compounds, monomers. They are so small, they cannot be seen with our naked eye. But if we use, for example, scanning electron microscope to look at the most stable aggregates, we can see these molecules. A very simple structure. And also, we also found that in the soil, those chemically complex materials, for example, plant lignin, these very complex materials, they actually can turn over fast in the soil. They can actually turn over very fast. Previously, we thought these chemically complex materials can stay in the soil for longer than simple structured materials. They can persist longer, and they contribute to soil organic matter formation. But now we have found that these plant lignin, they actually can turn over very fast, or faster than some of the microbial derived simple sugars. So the microbial derived simple sugars, although they are very simple in their structures, but sometimes they can stay in the soil for very long times, for decades or 100 years. Why is that? Now, so in the last decade, there are some new conceptual models, conceptual frameworks, or evidence started to show that soil organic matter turnover is really not governed by recalcitrance. It's governed by accessibility, or the physical interactions. So, for example, this paper here, it showed that in the soil, the physical structure or physical protection is what keeps the soil organic matter or the compound stable. And also all carbon input into the soil will eventually be cycled, regardless of their chemical complexity. So even the most chemical complex materials, they can be cycled. So it's actually the physical protection that keeps the organic matter stable. For example, here's a visual illustration of soil aggregation. So we can see those brown material, they are covered, they are protected by the minerals, by the small aggregates. And because of this physical protection, they can be protected against the microbial degradation. And because of these new evidence and advancement, and we started to understand that the soil microbes, they actually play very important roles in contributing to soil organic matter formation and soil carbon sequestration. In 2017, Lan and co-authors published this paper to propose the term soil microbial carbon pump. So soil microbial carbon pump framework was a conceptual framework to demonstrate the importance of soil microbial activities in soil carbon sequestration. So according to this framework, we can see that all the carbon inputs from the plant materials, from roots or from the exudates, from roots, from litter, from stems or leaves, they will be processed and utilize the microbes. And that's the microbial necromass, or the microbial dead biomass that can form because of their simple structure and close proximity with the soil minerals. They can form the organo-mineral associations and then be physically protected in a stable soil carbon pool. So these microbial products include the extracellular metabolites, microbial necromancer. Of course, in a stable carbon pool, there are some plant-derived carbon as well. For example, some simple sugared plant root exudates. They are very simple structured. They can also directly form association with minerals and then be protected, contributing to the stable carbon pool. And this is described by the ex vivo modification process. The ex vivo modification process is a process that's also facilitated by the microbes, because to partially decompose or to modify these plant carbon inputs, we also need the extracellular enzyme. These extracellular enzymes are also produced by sort of microbial activities. So microbial activities produce these enzymes and release them into the soil. These enzymes can catalyze the decomposition or partial decomposition or modification of these plant carbon and cause the large compound to start to break down and go into the soil carbon pool. And the in vivo turnover process, indicated by the red arrow in this figure, showed that the carbon that can be directly utilized and synthesized by the soil microbes or the soil microbial carbon pump. And the microbes, they use this carbon as their energy source to grow their biomass, to reproduce. And when they die, and the microbial dead biomass or the necromass can form association with the minerals to go into the stable soil carbon pool. And according to some new estimation or modeling, now we start to realize that the microbial contribution to soil carbon pool might be larger than we thought. Yeah, historically, we only knew that so microbial biomass was about less than 5% of the total soil carbon pool. And now we start to realize that the microbial necromass or other microbial metabolites could contribute to as much as 60% or 80% of the total soil organic carbon pool. So that's way more than what we thought before, and can be much larger than the direct contribution of plant-derived carbon to the total soil organic carbon pool. And because that, to build stable soil organic matter, those carbon that can be protected in a mineral association by the organic mineral associations, to build these stable soil organic matter or soil organic carbon pool and enhance soil carbon sequestration, what we should do is really to promote microbial growth, right? We want to produce more microbial biomass and microbial necromass to have accelerated microbial cycling. So more microbial necromass can enter the soil and form association with the minerals to contribute to the formation of stable soil organic carbon. Of course, when something happens, for example, when plants need more nutrient from soil organic matter, or when tillage happens, this protected soil organic matter or the microbial necromass in organic mineral association, these protected carbon can also be liberated to be released to contribute to soil fertility, nutrient cycling, right? But what we really need, what we need is really accelerated cycling to have continuous carbon input to stimulate the microbial growth all the time, to keep the cycle going, to keep the pump working. So this really explains the difference between the organic and conventional agriculture. So in a conventional agricultural systems, synthetic fertilizers are used. For example, corn, soybean systems, usually we put in about 150 pounds per acre nitrogen for corn, right? This nitrogen is used to feed the corn, the roots directly. And because of that, the plant roots, the corn roots are very shallow. And they don't need to go very deep into the soil to mine the nutrients protecting the soil organic matter. They can just take up any nutrients from the synthetic nutrient fertilizer, nitrogen fertilizers. But in organic agriculture, we don't directly put in those soluble fertilizers to crops. Yeah? We grow cover crops to replenish the soil organic matter. We put in organic fertilizers such as compost and manure. Then these carbon or nitrogen, they are in organic form. And microbes, they love these organic matter, so they will use this organic input and grow their biomass and start to build more stable carbon pool. And when plants grow, for example, the corn, when the corn need a lot of nutrient, nitrogen in the peak growing season, they will use different mechanisms. For example, form association with mycorrhizal fungi and to work with the microbe in the soil to mine and to mineralize those organic matter to get their nutrients. So that's a very different mechanism between organic and the conventional agriculture. That also shows that the differences in the microbial activity and the microbial growth in the conventional organic agriculture systems. That's probably why we always see that organic agriculture tend to increase soil organic matter. But in the conventional agriculture, usually we don't see that happen. So there's something we can manage if we know that microbes are key to contribute to increase the soil organic matter formation and carbon sequestration. So microbial carbon use efficiency, it's a term to describe the efficiency of the soil microbial communities in using carbon substrates, For example, when the microbes get some carbon input, if they put more carbon input into their biomass to grow their biomass, then we say that the microbial communities are more efficient or high CUE. They can put more carbon that they eat into their biomass and less carbon released through respiration. So they are more efficient. And in some conditions, some soil microbes, they put less of the carbon input into their biomass and respire a lot of carbon into the atmosphere. This is a situation where soil microbes are less efficient or low CUE in utilizing the carbon input. So the microbial carbon use efficiency or the CUE is really about efficiency of microbial growth or biomass production versus the energy cost or carbon respiration. So how do we improve or enhance the soil microbial carbon use efficiency or the CUE? We want to provide the microbes with the food they like to eat. When the microbes have the food they like to eat, they tend to grow more carbon into their biomass and the less carbon respired as CO2. So what do microbes like? They like simple structured or the low carbon to nitrogen ratio materials, for example, the root exudates, the legumes, compost, and manure. These materials have low carbon to nitrogen ratios, for example, 10:1, 15:1, or 20:1. And because of that, they can better meet the requirement, the stoichiometric requirement of the microbes. So microbes tend to have higher microbial carbon use efficiency. But if you apply things like high carbon to nitrogen ratios, such as corn stover, wheat straw, these materials usually have very high carbon to nitrogen ratio, 60:1 or 80:1. Then microbes are stressed. They don't feel comfortable. They have a hard time utilizing these carbon contained in the material. So they show low microbial carbon use efficiency. That's really because the carbon nitrogen ratio in these materials are too high. And in the microbial biomass, the carbon to nitrogen ratio is about 8:1. So we really want to be close to that ratio so microbes can efficiently utilize the carbon input. So here's an example how microbial carbon use efficiency can be affected by agriculture systems or by the food they eat, and how that can be related to the carbon content or carbon stock in agriculture system. Here's a very interesting study published by Cynthia Kallenbach in 2015. So they looked into the two systems in the Kellogg biological stations in Michigan. So one conventional system and one organic system. We can see that the organic systems have higher soil organic carbon content than the conventional systems, right? But if you look at the MCP, the above ground net primary productivity, or the carbon produced by these system, you can see that the total carbon produced, above ground carbon produced, is actually higher in the conventional system than the organic system. The conventional system produced about 1,500 gram per year dry biomass, above ground dry biomass. But the organic system produced about 1,300 lower above ground dry biomass than the conventional system. And their rotations are the same. The only difference is that the organic system had clover, the clover cover crops. The clover cover crops, we know that the clover is a legume and it has low carbon nitrogen ratio. So that's the difference between organic conventional system. And then they also looked at the carbon use efficiency of the soil microbes by the C13 tracing method. They found that at 3 different time points, the organic system all tended to have higher soil microbial carbon use efficiency than the conventional system. And the correlation analysis also shows that the soil organic carbon content is positively related to the soil microbial use efficiency. So that really shows that the more efficient the microbial carbon use in the organic system, because of the inclusion of the high quality or low CN ratio plant input from the clover cover crops, can contribute to the carbon sequestration and more carbon in the organic system than the conventional system. So that's one way that we can manage agriculture systems to sequester carbon. We can manage the microbial efficiency. How can we do that? We can provide the right food to the microbes to stimulate their microbial carbon use efficiency, to help they grow more biomass, and to facilitate the formation of the organomineral association, to protection of microbial necromancer. The other thing we can do is to promote the microbial necromancer or microbial biomass production, and also the soil carbon sequestration as a result of enhanced microbial carbon use efficient microbial carbon pump, is that we can try to create an environment, a physical structure that's favorable for soil microbial necromancer stabilization. For example, here, this paper by Kravchenko et al. Also studied the research in Michigan. They proposed a term, the microbial spatial footprint, as a driver of soil carbon stabilization. So the microbial spatial footprint is the volume of the soil matrix that can be immediately influenced by microbial decomposition products. So that means that the soil matrix, the soil space, that can be influenced by the microbial carbon, where microbial carbon can be stabilized, right? And they found that the soil pores, the pore structures, are essential to carbon accrual by promoting the stabilization of microbial products. Soil pores are very important. As I mentioned, that you want 50% of the soil space is filled with pore spaces by water and by air, right? That's where microbes can live and reproduce. They also showed that the management system can really lead to the different physical structure or pore spaces, pore structures of soil that can impact on microbial spatial footprint. For example, when you have monoculture system, you have only one species, or very simplistic system, you have very few species, and they use very simple root channels. And then in this condition, that's what's been shown on the left-hand side of the figure, is that the soil doesn't have very big pore spaces. And if you grow more multi-species crops, you have diverse crop rotation where different root channels can impact the pore structure, the soil physical properties. Then you have more diverse the root architecture that can impact the soil pore spaces. Then that's where you can really enhance your microbial spatial footprint, that the microbial necromass can be stabilized and form association with the soil minerals. So we want more of these pore spaces, those micropore spaces in the soil. And this can be affected by the different crops you are growing in the soil. So monoculture in this case is not very helpful. But if you can rotate your crops, you can have more diversity in the system, then it's gonna help you to build a structure that will help microbial necromass that can be stabilized. Okay, here are also some examples to show that the microbial carbon pump can be managed to increase soil carbon. So this is from the Wisconsin Integrated Cropping Systems trial, where they had two systems where the different diversity of the prairie species are grown for 9 years. So on the left-hand side, this system had 6 species, including 2 grasses, 2 legumes, and 2 forbs of the prairie species. And on the right-hand side, it's a high diversity perennial, which had 3 grasses, 4 legumes, and 17 forbs, and 1 woody shrub. So much more diverse than the system on the left-hand side. So we can see that after 9 years, and the soil carbon stock on the left-hand side is about 23.4 metric ton. On the right-hand side, soil organic carbon stock is about 25.96 metric ton. So the additional soil carbon content in the high diversity prairie versus relative to the low diversity prairie is about 2.57 metric ton. And out of the 2.57 metric ton carbon, the additional carbon, there is about 92 about this carbon is of microbial origin, or is from the microbial necromass carbon, which is 2.36 metric ton. So that shows that the additional carbon that's really contributed by the microbial necromass. Another example, a study was from the University of Illinois Energy Farm, where they compared the 3 different systems and transition 3 different systems transitioned from the continuous monoculture corn from 2008 to 2014. And we can see that the first system on the top, that's actually the same system, the maize, and soybean system. So we can see that from 2008 to 2014, the soil organic carbon stock actually decreased from 15.21 metric ton to 14.72 metric ton. So there was a net decrease of soil carbon of 0.48 metric ton. And when we switch from the maize, soybean system to a switchgrass, a monoculture switchgrass system, we can see that from 2008 to the year 2014, there was an additional 2.67 metric ton soil organic carbon being sequestered after the transition to the switchgrass system. And we know that switchgrass grows very fast. They grow a lot of biomass, right? And in this additional 2.67 metric ton carbon sequestered after switchgrass was introduced, about 76% of that carbon, additional carbon, was from microbial necromass. That means about 24% was not from microbial necromancer mass. They might be from the plant-derived carbon, which was described as the ex vivo modification process by the microbial carbon pump framework. And on the bottom, in a mixed prairie system, when we switch from the maize soybean system to a mixed prairie system, we can see that from the year 2008 to the year 2014, there was about 2.34 metric ton carbon sequestered in the system. So that's not as much as the switchgrass system. We know that switchgrass system, grow a lot of biomass, right? So that's not surprising. But in the mixed prairie system, in the additional 2.34 metric ton carbon, about 93% of this carbon was from microbial necromancer. So that means when you transitioned to a perennial, diverse prairie system, then that's where the microbial carbon pump start to work and sequester carbon. That's where the microbial necromancer became the majority of the carbon being sequestered in the system. So these results really show that to utilize the microbial carbon pump framework to promote carbon sequestration, we should try to promote the production of microbial carbon. So we can achieve that by providing the right food to the microbes, what the food they want, right? Don't feed them endless corn year after year, corn residues, right? We can promote the formation of organo-mineral associations. We can also try to reduce the physical disruption to the formed organo-mineral associations. And at Rhodell Institute, we also started to assess the microbial carbon pump capacity in our long-term farming system trial. Here at Rhodell, we have the farming system trial started in 1981, is the longest running side-by-side comparison of organic and conventional systems in North America. It started in 1981, we had 3 major agriculture systems. A conventional system that's of simple corn and soybean rotation. A 4-year organic legume system that only relies on organic legume, the leguminous cover crops as the source of soil fertility. Apart from leguminous cover crop, organic legume system does not receive any input. We also have an organic manure system, which also utilizes composted manure as the additional source of fertility, apart from the cover crops. And after several decades, in 2008, we divided all these 3 systems into till and no-till. So in a conventional system, no-till was achieved by using GMO seeds and more chemicals, Roundups. And in organic system, the no-till was achieved by adopting the cover crop based organic no-till system. What we do is to plant a cover crop in the fall, and in the next spring, we terminate the cover crops by tools like the roller crimper. And the cover crops will be rolled down, and then the residues will form a mulch to suppress weeds in the growing season. So we don't have to cultivate multiple times to control weeds. So we start to look at the carbon content in these systems. So you can see that from 1981, the soil organic matter content increased gradually in the organic two organic system, especially the organic manure system, but remained largely unchanged in the conventional system. But when we look at the organic matter content in these systems divided by till and no-till, we can see that in the conventional system, that no-till did not really improve the soil organic matter content or soil organic carbon content. Remember, that's a very simple corn soybean system. So the food was not something the microbes love. But in the organic legume system and organic manure system, adopting no-till and help to tend to increase the soil organic matter content in the organic system. And when we put in a cover crop in a conventional system, cover crop only increased or tend to increase soil organic matter when tillage was used. So that's what we saw in the difference of the effects of no-till in organic and conventional systems. In a conventional system, adopting chemically based no-till did not really improve soil organic matter. Actually, it caused more severe compaction and herbicide-resistant weeds. So we are currently working on that, and hopefully by sometime later this year, we can get something published and to update to all of you. And also, we start to look at the microbial carbon pump capacity, look at the amount of microbial necromass in soil organic carbon in these different systems. And hopefully sometime maybe in the first half of 2022, in my next webinar, I can update you with the latest result from that aspect of research. So with that, I want to wrap up my presentation. And thank you for all your attention. And now I'm gonna open up for questions. Okay. So there's a question from Julia. Julia asked regarding the synthetic versus organic fertilizer cycles. You mentioned that the plant roots have to actually grow deeper in the soil to mine for the nutrients, etc. What is the benefit of deeper, longer roots opposed to the shallow roots in the synthetic fertilizer plants? Thank you, Julia. That was a great question. So if you're from the Midwest, from Iowa or some other state in the Midwest, you probably can remember last year in 2020, maybe in July, there was a very huge storm, unprecedented storm, right? That really flattened a lot of the cornfields. Then farmers ended up collecting crop insurance, right? So that's the result of having shallow roots. So in that case, the corn or other crops were only fertilized by the synthetic fertilizers on the soil surface. They don't have to grow very strong roots. They grow very fast in the early season. For example, here now in July, we have in our farming systems trial, we can see that in the conventional system, the corn looks much higher, much taller than the corn in the organic system. There's several reasons. First reason is that the corn in the conventional system was planted earlier in April, and because their seeds were coated with pesticides. So we could plant the conventional corn earlier in April, not afraid of the seeds being damaged by pests because their seeds are coated with pesticides. But in organic system, organic corn were planted in June because organic seeds obviously are not coated with pesticides. So farmers usually wait until the temperature is warmer to plant their seeds so the seeds can quickly germinate. And so when we have deeper roots in the soil, then the plants are obviously very much stronger than when you have shallow roots. And also, although they grow slower than the conventional corn, but if you come later in August or in September, you will see they will catch up. They will catch up, and because they get their nutrients or fertility from soil organic matter, from the plant microbe interactions. So they usually incentivize the microbes to get their nutrients. And because of the reproduction of microbial biomass, then we see an increase of microbial necromass. That's how carbon sequestration is possible in the organic systems. Okay, we have a question from San Diego. How is related soil organic matter with nutrient available in the soil? Well, soil organic matter contains carbon, nitrogen, phosphorus, sulfur, and other nutrients, right? So more soil organic matter means more inherent soil nutrients that can be supplied to the plant Of course, in the conventional agriculture, people thought that they could, just bypass this process. They can just apply anything that plants, the crops want by applying fertilizers. So that's different philosophies in organic and conventional agriculture. San Diego has another question. What about the mixed cover crops, soybean rotation without corn or wheat? Mixed cover crop soy rotation without corn and soybean. Sorry, I don't get that. Can you type again? Okay, Darren has a question. How deep is the soil profile is their active microbial community? That's a great question. So we compare organic and conventional system, right? So we see that in a conventional system, in the top 10 centimeter depth, top 10 centimeter, the difference between organic and conventional is kind of small. But when you go deeper, you really see a big difference. For example, in 20 to 30 centimeter depth, you see the microbial biomass or the soil organic matter was much higher in the organic system than the conventional system. So that's really the difference between organic conventional system. Is deeper in the soil. It's more significant in the deep soil. Okay, in the chat box, we also have something. Angel was asking if we give certificate for the webinar. Sorry, we don't do that. And Thomas asks a question about, is there any difference how we apply manure, organic fertilizer? Well, the organic manure, the application was just a principle. So each farmer, each individual can be doing things a little bit different, right? Based on their operations or their systems. So here, we only apply manure every 5 or 6 years. We apply that before oats to the oat in a rotation. But some of our neighbors, they're organic farmers. They follow a shorter rotation. They don't have oats or wheat in the rotation. For example, they only do corn and soybean. They tend to apply manure every year. We don't do that. We don't recommend that because, first of all, we want to advocate for more diverse crop rotation. And also, if you apply manure every year, you're going to have a big problem with phosphorus. So you have phosphorus being accumulated in the soil that can inhibit the assimilation of other micronutrients. And also phosphorus runoff can be a problem for the environment. And Jonathan asks, a slide showed that higher above ground biomass in a conventional system versus organic, although sequestration might be greater in latter. Lower yields are contrary to farming objectives. If I'm interpreting this correctly, how does one merge to opposing goals? Well, so I think what Jonathan was asking was that, do organic systems always have lower yields? I don't think so. What we found in our long-term farming systems trial is that in the first 5 years, from 1981 to 1986, our organic system had lower yields than conventional. That was true. That was the first few years when you transitioned from fields that have been managed or conventionally for decades. But since 1981 until now, after the first 5 years, when the soil microbiology, when the soil organic matter are restored, then we don't see a significant difference in yield between organic and conventional system. For example, last year, 2020 was the year we had corn in all these three systems, organic manure, organic legume, and a conventional system. We see that organic legume system, being a zero input system, yielded a little bit less than a conventional system. But organic manure system yield more than a conventional system. That was typical. And in the years of drought, in very dry years, organic system can produce about 30% more yields than the conventional system, right? So that's more resilient on the climate change conditions. And what's more is that when we look into the nutrient quality, for example, in 2020, we, apart from the yields, corn yields, we also looked into crude protein, vitamin B6, B3, B9. All these, crude protein and these vitamins were higher in the organic manure system than the conventional system. And they have similar amount of yield, right? Similar amount of yield and more nutrient dense product in the organic systems. So we think that's better for human health, better for the nutrient, but for the planet. Becky asked a question: if carbon is more stable in necromass, should we be focusing on increasing necromass instead of soil organic carbon content of given field pasture? When thinking about carbon credits for farmers and ranchers, should necromass be measured? So right now, necromass is still kind of a new measurement. It's expensive to measure. It's very time consuming. So I don't think it will be a standard measurement of necromass in the near future. And also, as I said, carbon content, or necromass, they are always being cycled. They're always being cycled. So what's more important is a system that can facilitate the continuous production of these necromass and the carbon, not just the absolute amount, right? So I think that's what's more important for maybe carbon credit. I personally believe we should, incentivize the system, the practices. Thomas asked, should we insert somehow some legume into the soil? Well, how do we do that? How do we insert these legumes into the soil? I think we should grow more. We should try to grow more legumes in our diverse species mixes. We should incorporate these leguminous species, that's for sure. But we should do that in a biological way to let them grow, let them establish the link with the soil microbes. Jacob asked a question: can you get same yield with regenerative system and conventional, and will it depend per crop area and the climate? I think that the answer to that question is an absolute yes. We can grow same amount of yield with the regenerative system and the conventional. And regenerative systems must be size specific and context dependent. For example, what we are doing here in Kutztown, Pennsylvania are not what we are doing in Georgia Center, not what we are doing in California, right? For each of these sites, we need to design specific, site-specific regenerative system that fit into their climate, their soil type, and their socio-economic context, right? And can we get the same yield with regenerative system as the conventional? Of course, yes, as we have shown that. But we need to remember that organic and regenerative system and conventional system are working very differently. The conventional systems usually are very simplified. You just put in input, chemical input, and ignoring all the biological interactions and ignoring all the detrimental environmental effects, consequences. And you manage thousands of acres, large scale by one person. While the regenerative system is a knowledge-based system, it needs more effort, more knowledge, but definitely it can be profitable and productive. What cash crops are recommended if the goal is to increase carbon in necromass? Well, I think that's a question from Becky. Thank you, Becky. That's a great question. Well, that's really something we need to think about economics and food demand and many other factors. For example, currently, why do farmers grow so many corn and soybean acres? Why? It's because that the system, this incentivizes them to do that. And in different parts of the world, it can be a little bit different. For example, in the Midwest, in large areas of the Midwest of the United States, the soils formed under the prairie, the tall grass prairie. So I would think to facilitate the carbon sequestration or microbial necromancer production, we should try to return much of that land to prairie, to perennial prairie, grassland, grazed rotationally by large herds of maybe dairy cows or cattle, because those soils form the prairie. Those prairies were grazed by bisons, large animals. That's how that rich molecules form in that context. But here in the Northeastern United States, we don't have that much, prairie or large herds of animals, bisons. Soils here formed under the Northeast forest. We have more precipitation here. So maybe we should try to do more agroforestry here. So this really needs to take into account of the different factors. But I think ideally we want to have a diverse species, whatever it is in different locations, or grasses or the tree species. We want to have soil covered to have more plants growing all year round. And we also want to minimize the disturbance to promote the formation of that organomineral association and the protection of the microbial necromancer. Okay, Toby from Germany, organic vegetable farmer. Hello, Toby. You said low carbon nitrogen ratio inputs such as legumes improve microbial carbon usage efficiency, but high carbon nitrogen ratio decrease carbon usage efficiency. Does this mean we should always try to minimize the input of high carbon nitrogen ratio materials, or only as the start to get the microbes going, then later we can add more high carbon nitrogen ratio materials? In practical terms, is it advisable to grow cover crop with a lot of rye and then grow cover crop with a lot, then crimp them? And should we switch to cover crop that have more legumes? Okay, that's a great question. Have you tried compost? You probably have done compost in your backyard, right? So in composting, usually we recommend the beginner to use some brown material and some green material, right? The brown material are more high carbon content material, those leaves, those barks, those different mulches, different things. And the green materials are the food waste, or the green leaves, or these type of things. We want a mixture of these things. Then we can promote a balanced community structure of microbes, including fungi and bacteria. So I wouldn't say we only grow legumes to grow more carbon. I think we should try to mix different things. And that's just a principle that if we have an ideal carbon nitrogen ratio, maybe around 20:1, then we can maintain very efficient microbial communities. But to achieve that, we can have some, rye in the soil profile, we can have some legume in the soil profile, then we have a good mixture. Also for cover crops, we grow different cover crops for different purposes. For example, we grow rye before the soybean. Why? When we roll down the rye, they can really form a very thick mulch for soybean. Soybean needs a lot of protection from weeds. That's why we need a lot of biomass. That's what rye does, right? They grow a lot of biomass. And when you roll down the rye, they can really protect the soil, protect soybean against the weeds. But for corn doesn't need very, much, protection from weeds, because for corn, they can close their canopy very fast. They can outcompete weeds very effectively. So what corn really needs is nitrogen, fertility. What really constrains corn is fertility. So that's why we usually grow hairy vetch or clovers before the corn, because they can provide nitrogen credit to the corn. If we grow rye before the corn, then rye will tie up nutrient cycling, will cause nutrient deficiency, nitrogen deficiency of corn and cause yield decline. Jonathan asked, is biochar considered as mineral fraction that is associated with reduced compact decomposition? Or does biochar decompose also, or it is not appropriate for long-term carbon sequestration? Biochar is more stable. Yeah, it's not something that microbes can, actively, utilize in their in vivo turnover process, because they have been through that combustion process. Usually they just add carbon content into the soil, but the cost for biochar production and the viability for biochar production, that's something, if we can achieve that at a low cost on a large scale, I think that has some benefits. Pat asks, Necromancer spelling, so it's Necromass, Necromancer. Okay, we have some more questions in the Q&A. Okay. Okay, Johannes asked a question. Do you know of any successful mixed cropping system that imitate a prairie or similar, but with annuals? Which crop would be suitable? Sorry, I don't think I can recall that information. And Santiago asked, what happens if we only do cover crops, soybean, without including corn or wheat in the rotation? Oh, you mean only do corn, cover crops and soybean? Of course you can do that, but again, it's a monoculture system. You have less diverse type of carbon input. Then you have limited, capacity to incentivize different types of microbe in the soil. So I would say that corn is okay. Planting corn, including corn, is okay, but don't grow them every year. Same for soybean. Johannes asks, what about the importance, the role of fungi in microbial carbon capacity, carbon pump? That's a great question. So we want a balanced structure of soil microbe in a microbial carbon pump, right? And fungi and bacteria, they have different roles. Fungi, they can produce the oxidative enzyme that can catalyze the decomposition of complex plant materials, these lignins. They can make them smaller and feed the bacteria. And also fungal necromancer are usually thought to be more stable in the soil organic matter. Carol asked, if necromancer is a key to soil organic carbon, does that mean that open animal burial grounds have exceedingly high soil organic carbon levels? Well, I think the key is living things. Animals have always been part of the system. Animal has always been part of the system. As I mentioned, for example, in the high tall grass prairie in the Midwest, we have those tall grass prairie growing, right? They have a lot of above ground biomass, but also they have very big amount of below ground biomass that can go several meters deep. And those large herds of animals, what they do is not just feeding the microbes, they also help the plants grow. For example, when they graze the tall grass prairie, they can accelerate the regrowth of these plants. They can also accelerate the below ground growth. So that's efficient animal, plant, soil, and micro interactions. The systems co-evolved together. Nothing is ignored. So we need the system to be functioning to promote soil carbon sequestration. West asked a question, are the Rhodell long-term studies available to view? If so, do you have a link? Yes, please go to our website, rhoddellinstitute.org, and you can go to research. You can also access to our peer-reviewed publications, our web articles. We have a large database of these information that's available for you to review. Okay, I hope you all enjoyed today's webinar. I hope you have learned something. So please stay in touch. And next time in September, Dr. Drew Smith, our Chief Operating Officer, will be updating you with our newest research result from the nutrient quality grown in different agriculture systems. So please register for that webinar. And if you have any questions, please send your email to info@rhodellinstitute.org and mention soil microbial carbon pump webinar. And I'll be happy to answer your questions. And with that, I want to thank you again for your attention and hope to see you soon. Bye.